Good morning, students. Myself, Mohammad Imran Anis, Assistant Professor, YB Chavan College of Pharmacy. Today we are going to see about the parts of microscope and their function. If you see over here, this is the construction of a microscope. We will go one by one with the parts of microscope. Now if you see over here, the first part, it is the tube. This tube, it is reflecting the light up to the viewer's eyes. Now this tube, it will get the light from the objective lens, okay, and then after this we can visualize the light through this particular draw tube. It reflects the light up to the viewer's eyes, that is the eyepiece. If you see over here, this is the second one, that is called as the revolving nose piece. This revolving nose piece, it allows to quickly change the objective lens according to the specimen which you have to adjust. On this screen, if you see, this is the third part, that is the low power objective lens. Low power objective lens, the first lens which you use when doing the microscopic working. This low power objective lens, in different microscope, different low power objective lens are there. Usually over here, if you see, it is of 4x, but in different microscope it will be 10x also. Then medium power objective lens, over this particular slide, this fourth one, the second objective lens which you are using when doing the microscopic work, it is of 10x, okay. In medium power objective lens, it is 10x. Then the high power objective lens over here, if you see, this is the high power objective lens which is of 40x or 45x. Apart from this high power objective lens, there is one more objective lens in a microscope, that is called as 100x. High power objective lens 100x, it is also called as oil immersion objective lens. It is 100 times having more magnification power than the other objective lens. So, but the only thing which you have to need to understand is that 100x objective lens, it is oil immersion objective lens, and in that the oil has to get been dipped in the objective lens. Means the slide, on the slide you have to add a single small drop of oil for magnification power, and then only you can see the image in the eyepiece. 100x, that is the oil immersion objective lens. The oil which you have to use is cedar wood oil. Then the sixth part, if you see over here, these are the stage clips. These stage clips, they are used to keep the slides in proper place. When you have to adjust the slide, you have to place it under this particular stage clip. Then the seventh one over here, below the stage, you are observing a diaphragm. This particular diaphragm, it is used to vary the amount of light passing through the slide. And in this particular diaphragm, it is also called as iris diaphragm, IRIS, iris diaphragm. This iris diaphragm, it will magnify the light and then pass through the condenser. On this particular diaphragm, there is placement of condenser which will focus the light upwards towards the specimen. The light source, if you see over here, the light source which you are visualizing, this sends lights through the diaphragm and then through the slide for viewing. Over here, the light which has been used is the artificial light, but in normal compound microscope, the light source which has to be governed, it is been placed by having mirror in it. The light source can be captured by mirror, and this mirror, they have two lenses. One is concave lens, concave mirror and the other is the flat mirror. Concave mirror, it is used for the natural light, and flat mirror, it is used for the artificial light. Now in this particular slide, if you see, ninth one, it is the eyepiece. This is the eyepiece, the part of the microscope where you are placing your eye, and on this eyepiece you can visualize the image which is coming from the specimen. This is the neck, 10th part. This neck, from this particular neck you can safely transport the microscope. You can handle the microscope by use of this particular neck. Stage, this is the stage. The slide is placed on this stage and it has been attached on this particular clips, that is the slide clips or stage clips. If you see over here on this particular part, this is the coarse adjustment knob. This coarse adjustment knob, it is used to make changes in the focus, and coarse adjustment knob, you can change the focus of the image or the magnification process. Fine adjustment knob, over here you can see the fine adjustment knob. It is used to adjust the slide specimen properly, and you can visualize the image in the slide by properly focusing the fine adjustment knob. The last part, it is called as base. If you see over here, this is the base. It is used to safely transport the microscope from one part or from one stand to other stand. So this is all about the parts of microscope and its function. Remember students, while handling the microscope you have to take care and you have to clean the microscope before and after use. Before and after use you have to clean the microscope, specifically the objective lens, the eyepiece and the mirror, with a clean soft cloth or a clean soft tissue paper. You have to clean it properly to avoid the dust particles to enter into the microscope. Cleaning of the microscope properly will enhance the shelf life of the microscope and you can visualize the image properly. So this is all about the parts and functions of microscope. Thank you very much for your patient listening.